A number of reagents used in molecular cloning techniques are toxic, hazardous, radioactive, and potentially carcinogenic. The individuals performing such experiments must understand the safety considerations and carry out laboratory experiments with maximum precautions. This videotape should be used exclusively for understanding the concepts of DNA cloning techniques. Such experiments must be performed after going through detailed protocols. Science Vision International, or Biotech Visions, assumes no liability for any injury resulting from the performance of such experiments. Molecular cloning of DNA provides a mechanism for isolating a single segment of DNA from a population of millions of genes. These genes are used for genetic and biological analysis and can also be introduced into cells or whole animals to study the function and regulation of wild-type or man-made mutant genes. In the past 10 years, DNA cloning research has undergone dramatic expansion. DNA cloning techniques have succeeded in making hundreds of gene transplants into the cells of higher organisms. The tape you're about to see contains 2 volumes. In Volume 1, the following subjects will be covered. First, a brief introduction, followed by restriction and modifying enzymes, bacteriophage infection, genomic library, plasmid preparation, gel electrophoresis, DNA subcloning, and DNA hybridization. Then, in the second volume, we will continue with southern blot applications, dot blot, DNA sequencing, mutation, polymerase chain reaction, DNA footprinting, S1 protection assay, polyacrylamide gel electrophoresis, and western blot. The genetic material of the cell, DNA, is a polymer composed of 4 nucleotide building blocks, which are adenine, guanine, thymine, and cytosine. In addition to these nucleotides, DNA also contains deoxyribose moiety and a phosphodiester backbone. Chains of these DNA subunits exist as 2 antiparallel strands in opposite direction, resulting in a 3-dimensional double helical structure. Each strand binds to another due to hydrogen bonding between these 4 bases. Adenine is always paired with thymine and guanine with cytosine. It is the specificity of this complementary base pairing that forms the basis of DNA's genetic information. In the synthetic pathway between DNA and protein are the ribonucleic acids, RNA. During the process of RNA synthesis, the protein sequence information of DNA is copied into a complementary strand of RNA. The process of RNA synthesis is called transcription. The RNA contains the same bases as DNA, except that uridine is substituted for thymine, and ribose moiety is present instead of the deoxyribose. Messenger RNA, which is a copy of the gene, is finally translated into proteins. The genetic information carried by an RNA molecule is converted into an amino acid sequence of proteins. The actual assembly of amino acids to form proteins involves the assistance and cooperation of transfer RNA and ribosomes. Here you see a 3-dimensional structure of a bacterial protein named REC-A. The protein is involved in genetic recombination and DNA repair. This protein contains 352 amino acids with a molecular weight of 38,000 Daltons. Coming up is a selection of about 16 terms and definitions essential for understanding the basic concepts of biotechnology. The nucleus of eukaryotic cells contains several chromosomes, which consist of 50% DNA and 50% proteins, mostly histones. Each chromosome contains a single large DNA molecule. The process by which DNA copies itself is called replication. DNA replicates so that every new cell receives a complementary copy of the genetic code. The process by which mRNA is copied from DNA is called transcription. Only one strand of DNA takes part in the transcription. Every combination of 3 bases of messenger RNA is called a codon. Such triplet codons enables the messenger RNA to collect the right amino acids for protein synthesis. The process by which the mRNA base sequence is transferred into the amino acid sequence is called translation. Small ring-shaped segments of DNA are called plasmids. Inside the bacteria, the plasmid remains separate from the bacterial chromosome. If plasmid has a gene for production of an enzyme that destroys penicillin, bacteria containing these plasmids become resistant to penicillin. Vectors share the similar properties as plasmids, except that vector DNA can be linear or circular. Transformation is an artificial process by which DNA is transferred into prokaryotic, such as bacterial or eukaryotic cells. Electroporation is the electrotransformation which requires an electric field to introduce DNA molecules into such cells. Bacteriophage lambda is a double-stranded DNA virus which contains linear duplex DNA. After entering the host bacterium, DNA circularizes and is transcribed as a circular molecule. A change in a gene or chromosome is called a mutation. Point mutation. One base of the DNA is substituted, lost, or added. Changing even one base in the genetic code can have major consequences. Genetic engineering. Genetic engineering involves the deliberate alteration of the genetic material of living cells. The altered DNA is called recombinant DNA. Molecular genetics. The study of the molecular structure of the genetic material, DNA and RNA, and the transfer of genetic information from one generation to the next is called molecular genetics. Gene. A gene is a stretch of a DNA molecule that acts as a functional unit for genetic information, such as genes for hair color, eye color, blood type, etc. There may be thousands of genes in a single DNA molecule. Genome. The entire complement of genes in an organism, regardless of their physical arrangement within the cell, is known as the organism's genome. Regulatory gene. The regulatory genes are the lengths of DNA whose function is to control the activity of other genes. Gene expression. Gene expression involves the study of the transfer of genetic information from DNA to RNA to proteins. Restriction and modifying enzymes. Restriction enzymes and other modifying enzymes are extremely important tools in molecular biology. They are used to cut, copy, splice, or modify discrete DNA molecules. SAL1, like all restriction enzymes, binds specifically to double-strand DNA and cuts at specific sites known as recognition sequences. SAL1 recognizes 6 bases of double-strand DNA molecule, and after cutting the DNA, produces 5' protruding ends. PST1 is another common enzyme which also recognizes 6 DNA bases of double-stranded DNA. Like SAL1, it also cuts in an axis of symmetry, but it produces 3' protruding ends. Some enzymes cut double-strand DNA, creating fragments of DNA without protruding ends. Here is an example of one such enzyme, HINK2. Notice here that after cutting this DNA, the fragments are left without either 5' or 3' overhangs. Therefore, they have blunt ends. DNA ligation can be carried out by the enzyme T4 DNA ligase. This enzyme joins the enzymatically digested DNA fragments. Shown here is an example of the ligation of enzymatically digested DNA having non-compatible ends. These pieces of DNA contain bases having the wrong base pairing. Therefore, these DNA ends cannot be ligated by the ligase enzyme. However, we can use linkers to make the ends compatible for ligation. Here, you can see a linker, which is a small synthetic oligonucleotide that is linked between 2 compatible ends of DNA. Now that there is the correct base pairing, these pieces of DNA can be ligated with T4 DNA ligase. Another common enzyme used in molecular biology is exonuclease 3. This enzyme results in the stepwise removal of 3' to 5' nucleotides from both ends of the double-stranded DNA molecule. Exonuclease 7 results in the stepwise removal of nucleotides from single-stranded DNA and leaves the double-strand DNA intact. Clinau is a very common modifying enzyme used in molecular biology to fill the 5' protruding ends of DNA. Similarly, T4 polymerase fills the 3' protruding ends of DNA. These two modifying enzymes, Clinau and T4 polymerase, can also be used to make radioactive DNA probes. Such probes are made by end-filling the DNA in the presence of radioactive nucleotides. Bacteriophage M13, a single-stranded virus which contains single-stranded circular DNA of approximately 7 kilobases in length. Soon after entering the bacterium, DNA is converted into a double-stranded DNA, which can be isolated, purified, and used as a DNA cloning vector. Bacteriophage lambda. Bacteriophage contains one double-stranded DNA molecule of approximately 50 kilobases in length. In the early stages of bacterial infection, one of the two pathways of replication are chosen: lysogenic growth. The bacteriophage DNA becomes integrated into bacterial host DNA. Lytic growth. The circular DNA is replicated many-fold, and bacteriophage particles are assembled, which results in bacterial cell lysis. In the lysogenic cycle of bacteriophage infection, the viral DNA gets integrated with the host DNA. This process does not lead to bacterial lysis. However, in another cycle, which is called the lytic cycle, the bacteriophage attaches to the bacterial cell wall and releases its DNA. Soon after this infection, the bacteriophage takes over the host bacteria, resulting in the formation of viral DNA, sheaths, heads, and tails. Thus, complete assembly of bacteriophages begins on the bacterial plasma membrane. Finally, bacteriophage particles break off from the host cell, resulting in bacterial lysis. These bacteriophage particles are used for the isolation of DNA. Genomic library. The first step in the molecular cloning of eukaryotic chromosomal DNA is to make the genomic library, which contains millions of recombinant genes. Genes of interest are isolated from such libraries for genetic and biological analysis. Major steps to prepare genomic library. Eukaryotic DNA of interest is prepared from cells containing desired genes. Genomic DNA is digested with appropriate restriction enzymes. Resulting fragments of DNA are introduced into appropriate vectors. Next, recombinant DNA molecules are packaged into infectious bacteriophage particles. Finally, hybridization techniques are used to pick up the bacteriophage particle containing desired DNA. The first step in making a genomic library is to select high molecular weight genomic DNA. This DNA, containing desired DNA sequences, is then used for partial digestion with an appropriate restriction enzyme. The desired vector DNA is selected where these genomic fragments can be inserted. The internal fragments of vector DNA are removed with restriction enzymes. Now the resulting vector is ready for ligation. The genomic DNA fragments are inserted into this vector DNA in the presence of the ligase enzyme. This results in the formation of recombinant DNA molecules. In other words, these are basically the vectors containing DNA inserts. The resulting recombinant DNA molecules are packaged to make infectious bacteriophage particles. This library, containing billions of such bacteriophage particles, is used to screen specific gene sequences. Plasmid DNA preparation. Several methods are used to extract plasmid DNA from bacterial culture. All of them involve three basic steps. Grow the bacteria containing desired plasmid, lyse the bacteria, and collect the supernatant containing plasmid DNA solution. Purify the plasmid by cesium chloride or DNA resin affinity methods. Here we present two different methods for plasmid preparation: the cesium chloride method and the DNA resin affinity method. First, we present the standard cesium chloride method. A single well-isolated bacterial colony is picked up using a sterile loop. This colony is transferred to a small sterile bacterial media containing an appropriate antibiotic. This media is grown overnight and then transferred to a larger media. This is again grown overnight, and the resulting growth of bacteria is transferred to a wide-mouth bottle. The bacterial media in this bottle is centrifuged to produce a bacterial pellet. At this stage, the supernatant solution is taken out and discarded. Now into the bottle containing the bacterial pellet, an appropriate amount of both bacterial lysis and neutralization solutions are added. After another centrifugation, this solution, which contains the plasmid DNA, is transferred into another tube. The pellet containing debris is discarded. An appropriate amount of cesium chloride and ethidium bromide are now added into this solution. This solution is now transferred to an ultracentrifuge tube, which is then sealed and spun into an ultracentrifuge. This procedure produces two bands, one containing the NIC DNA and the other plasmid DNA. The lower band of plasmid DNA is extracted using a hypodermic needle. This pink solution containing plasmid DNA and the dye ethidium bromide is now transferred into another tube. The ethidium bromide is extracted using water-saturated isopropanol. Finally, you are left with a solution which contains purified plasmid DNA. If necessary, this DNA solution can be further purified using ethanol precipitation. When using the DNA resin method, you take the bacterial lysate as prepared in the previous method and add DNA-binding resin. This resin has a special affinity for DNA. RNA and proteins won't bind to it. You then mix gently and allow the DNA to bind with the resin. This mixture is then transferred to a column which contains a filter at the bottom. A vacuum line is attached to the bottom of this column and a gentle vacuum is applied. This process extracts the impurities from the mixture, leaving only the resin bound to the DNA. This mixture is washed with ethanol, and the bound DNA is extracted with water. This extraction results in the isolation of the DNA from the resin. This purified DNA can be used for further analysis or subcloning. Now we present the live footage of the plasmid preparation using cesium chloride gradient method. Here we show the streaking of bacterial plates with desired bacteria. Bacteria are streaked near flame to avoid any undesired bacterial contamination. After streaking, these plates are incubated at 37 degrees centigrade for the bacterial growth. On the following day, bacterial colonies can be easily seen on these plates. A single bacterial colony is picked up for inoculating small bacterial media in a test tube. The media is grown overnight at 37 degrees centigrade with constant shaking. Shaking helps the growth of the bacteria. Otherwise, the bacteria would settle down and they would not grow properly. This small media is finally transferred to a larger media. Although the media in the large flask could also be inoculated with a single bacterial colony, adding already grown bacterial culture produces overall better bacterial growth. This media is now carefully weighed. The weight of media is properly balanced so that there is equal weight in these bottles. These bottles are then placed into a centrifuge rotor, and the media is centrifuged to produce a bacterial pellet. The supernatant is discarded, and the bacterial pellet is used for the preparation of desired plasmid. After mixing the bacterial pellet with an appropriate buffer, bacterial lysis solution is added. Sodium hydroxide solution is normally used as the bacterial lysis solution. Finally, a sodium acetate solution is added to neutralize the pH of the solution back to normal. Now this media is centrifuged again, and this time the bacterial pellet is discarded, and the supernatant containing the desired plasmid is used for plasmid extraction. Appropriate amounts of ethidium bromide and cesium chloride are now added into this solution. This solution is carefully poured into ultracentrifuge tubes, and then are heat sealed to avoid leakage of the solution. These tubes are inserted into the ultracentrifuge rotor for high-speed ultracentrifugation. Normally, a speed of 50,000 revolutions per minute is used for about 15 hours. This produces a gradient of cesium chloride, and the plasmid DNA bound to the ethidium bromide gives the discrete band, which can be easily visualized by the naked eye. However, this band becomes more prominent if seen under UV light. This plasmid DNA is carefully extracted by using a sterile needle and a syringe. Ethidium bromide bound to DNA is extracted by using water-saturated isopropanol. The top solution, which contains a mixture of isopropanol and ethidium bromide, is finally removed using a pipette. lower solution containing the desired plasmid DNA is used for ethanol precipitation. Here you can see the plasmid DNA precipitated by ethanol. After another centrifugation, the ethanol solution can then be discarded and the DNA pellet can be dissolved in water. Now we present the live footage of plasmid preparation using the DNA resin affinity method. In this case, the first few steps, such as bacteriolysis and neutralization steps, are the same as in the previous case. Once you have produced the bacterial lysate, instead of adding ethidium bromide and cesium chloride, a resin is added which has a special affinity for the DNA. This solution is passed through a column which has a filter at the bottom. The DNA bound to resin gets trapped in this column. The resin containing bound DNA is then washed using ethanol or isopropanol solution. Finally, the DNA is eluted by adding a small amount of warm water. Approximately 5 minutes of centrifugation at low speed is enough to extract this plasmid DNA from the bound resin. Here you see the extracted plasmid DNA in water. Plasmid DNA preparation. Special notes. Typical yield of plasmid DNA. It's 2 to 5 milligrams per 500 milliliters of bacterial culture. In general, the smaller the plasmid, the better the results. For plasmids greater than 20 kilobases in size, the isolation is very difficult and the yield is poor. Agarose gel electrophoresis, the standard method to resolve DNA fragments and determine their length by comparing them with fragments of known length. DNA is digested with appropriate restriction enzymes. Resulting fragments are loaded on agarose gel electrophoresis. Smaller fragments migrate faster than larger ones. Bands of DNA within the gel are stained with a fluorescent dye such as ethidium bromide. Since the introduction of agarose gel electrophoresis, many different designs of apparatus have been used. Here we see different sizes of sample loading combs, which are used depending upon the volume and the concentration of DNA to be separated by electrophoresis. Also, different concentrations of agarose are used depending on the length of the DNA fragments and the range of separation required. A correct amount of powdered agarose is mixed with a measured amount of electrophoresis buffer. This mixture is then boiled in a microwave to dissolve the powdered agarose. The agarose slurry is then transferred to the gel casting container. After the gel is completely set, which takes about half an hour, the comb is carefully removed and the gel is mounted in the electrophoresis apparatus along with its casting container. An appropriate amount of electrophoresis buffer is then added to cover the gel to a depth of about 2 millimeters. Samples are now mixed with a loading buffer and then loaded into the wells of the submerged gel. The maximum amount of DNA that can be loaded into the well depends upon the number of fragments and the size of the gel. Normally, 1 to 5 micrograms of DNA can be loaded per well. Finally, electrophoresis is carried out by attaching the apparatus to a power supply. This electrophoresis is run for a specific amount of time, depending upon the required separation of DNA fragments. The DNA in the agarose gel can be seen using a fluorescent dye, such as ethidium bromide. When this stained gel is exposed to ultraviolet light, the absorbed light is emitted, giving the DNA an appearance of red-orange colored bands. A photograph of the gels can be made using an instant laboratory camera. Here, you see a picture that was taken with a laboratory camera showing the DNA bands stained with ethidium bromide. Recovery of DNA from agarose gel. DNA from the gel can be recovered by any of the following methods: direct electroelution, dialysis bag electrophoresis, phenol chloroform extraction, and DNA resin affinity methods. Here we present one of the various methods for the recovery of DNA from a gel slice. After performing electrophoresis on the DNA samples, localize the DNA bands of interest under ultraviolet light. Using a sharp scalpel or razor blade, cut away the piece of the gel containing the desired bands of DNA. Insert this piece into a small microfuge tube. An amount as small as a few nanograms of DNA can be extracted and purified from these gel slices. Now a small amount of buffer is added into this microfuge tube, enough so that the gel slice is completely submerged. A small microfuge tube containing a filter is then inserted into this tube for electroelution. Electrodes are inserted into this tube and electroelution is performed using gel eluter apparatus. Electroelution is carried out until the DNA present within the gel is extracted and is suspended within the surrounding buffer. The empty gel slice is taken out and discarded. Special notes. Ultraviolet radiations are dangerous to the skin and eyes. The UV source must be properly shielded and protective glasses should be worn. Ethidium bromide is a potent mutagen. Proper care should be taken for handling this reagent. As little as 1 nanogram of DNA can be detected by the direct illumination of a stained gel with UV light. Very large DNA fragments, 10,000 kilobases, can be separated by pulse field electrophoresis, in which the electric field is changed periodically. DNA subcloning. DNA subcloning involves the ligation, covalent joining, of foreign DNA to desired vectors or plasmids by using restriction enzyme and T4 DNA ligase. Antibiotic-resistant genes within the plasmids are used as markers for identification of bacterial colonies containing recombinant DNA. Insertional inactivation of genes is analyzed by bacterial resistance to antibiotics. Molecular subcloning is carried out in microfuge tubes, as shown here. This is an example of subcloning, where a piece of foreign DNA is mixed with a vector DNA in the presence of the T4 DNA ligase enzyme. Now, in the upcoming animation, you will see several examples of DNA subcloning. First is the insertion of foreign DNA with compatible ends into a plasmid DNA. Here, you can see a double-stranded plasmid DNA, which contains 2 different genes, an ampicillin-resistant and a tetracycline-resistant gene. A piece of foreign DNA with compatible ends can be directly inserted into the ampicillin or tetracycline portion of the plasmid. As shown here, this insertion of foreign DNA into the tetracycline portion of plasmid results in the destruction of the tetracycline gene. Next is an example of the insertion of foreign DNA with non-compatible ends. In this case, the foreign DNA ends are not compatible with the restriction enzyme digested ends of plasmid DNA. In order to subclone this fragment of DNA, you must first attach linkers to make the ends compatible. Now the DNA can be ligated in the presence of T4 DNA ligase. The third possibility is the insertion of foreign DNA taken from a pool of several fragments. In this case, suppose you have a linear DNA molecule. This is digested with an appropriate restriction enzyme to produce different DNA fragments. In the example shown here, the DNA produces 3 fragments, which are then separated by agarose gel electrophoresis. These fragments are then stained with an appropriate dye, such as ethidium bromide. At this point, the DNA band of interest is excised using a clean razor blade and is used for DNA extraction and purification. Later, this DNA is inserted into an appropriate plasmid. Finally, such cloned plasmids are transferred into bacteria, which are then grown on bacterial plates. Here, you can see 2 types of plasmid DNA molecules. One is a wild type containing cloned DNA. The other is strictly wild type. Both of these are transformed into bacteria. These bacteria are then grown on tetracycline plates. On the next day, you can observe the plates for the presence of bacterial colonies. You can see that in the right plate, which had wild-type DNA, a number of bacterial colonies grow because the tetracycline gene was not interrupted. However, on the left plate, no bacterial colonies grow because the tetracycline gene was interrupted due to the presence of foreign DNA. In addition to tetracycline plates, these bacteria are grown in bacterial plates containing ampicillin. In this case, bacterial colonies grow in both plates, as the ampicillin gene was not interrupted in either case. Finally, you can pick the bacterial colonies from the ampicillin plate containing recombinant plasmid DNA and proceed with the DNA preparation. You can also confirm the presence of foreign DNA inserts by using restriction enzymes. Hybridization. This technique is based upon the base pairing of radioactive labeled DNA to complementary sequences of DNA or RNA molecules. Screening of bacterial colonies. Replicas of bacterial colonies are made. These colonies are lysed on nitrocellulose filters. Released DNA is allowed to bind to the nitrocellulose filters. Bound DNA is hybridized with the appropriate radioactive labeled nucleic acid probes. Positive bacterial colonies are picked up after autoradiography. The process of DNA hybridization is used when it is necessary to screen plaques or bacterial colonies on agarose plates. Nitrocellulose paper is laid on the surface of these agarose plates. Orientation marks are made on the nitrocellulose paper in asymmetric positions. Normally, 2 nitrocellulose papers are used, the second a replica of the first, but for simplification, only one is shown here. This paper, containing the impression of the bacterial colonies, is transferred into soaking pads containing DNA denaturization solution. After about 10 minutes, the filter is transferred to another soaking pad containing neutralization solution. This treatment releases the DNA from the bacterial colonies, and after baking at 80 degrees centigrade, this DNA binds permanently to the nitrocellulose filter. This filter is now inserted into a sealing bag. This sealing bag is heat sealed and then cut at the corner. This process makes it easier to add the following solutions. The bag is now filled with pre-hybridization and hybridization solution and sealed again to avoid any leakage of radioactive isotopes. Overnight hybridization with radioactive DNA is performed usually at 42 degrees centigrade in presence of 50% formamide solution. On the following day, the nitrocellulose paper is removed from the bag and used for autoradiography. This results in different black spots corresponding to positive bacterial colonies. Isolated positive bacterial colonies are identified on the agarose plate and picked up by using a sterile loop. These bacterial colonies are grown in a bacterial media for plasmid preparation. Here we present the live footage of the DNA hybridization of bacterial colonies. A nitrocellulose filter paper is placed on top of these bacterial colonies. This paper is later lifted carefully so that the bacterial colonies stick to the bottom of this nitrocellulose filter. Orientation marks are made on the top of these nitrocellulose papers by using a colored pencil. Such permanent marks are used later to align these filters with autoradiographs. Another wet nitrocellulose filter is laid on top of this nitrocellulose. By pressing gently, a replica of the bacterial colonies are made on this second nitrocellulose filter. This filter paper is now transferred to wet soaking pads containing different solutions, such as sodium hydroxide as a DNA denaturation solution and sodium chloride tris buffer as a DNA neutralization solution. These filters are then baked at 80 degrees centigrade for approximately 30 minutes. At this temperature, the DNA binds permanently to such nitrocellulose filter papers. These filters are then inserted into sealing bags for pre-hybridization and hybridization. Here we are showing an example of the DNA hybridization using radioactive DNA probes. However, non-radioactive probes can also be used for DNA hybridization. In that case, special handling of radioactive solutions and use of protective shields, as shown here, are not necessary. The bags containing nitrocellulose papers and hybridization solutions are gently shaken in a water bath for DNA hybridization. After overnight hybridization, the filters are removed from these bags and the radioactive solution is discarded properly. These nitrocellulose filters are then washed properly to get rid of any unbound radioactive DNA probes. Finally, these nitrocellulose papers are dried and then used for autoradiography. These autoradiographs are aligned with original bacterial plates, and the right bacterial colony is picked for its growth and the plasmid preparation. Special note, it is easily possible to screen thousands of bacterial colonies simultaneously to find positive colonies carrying desired recombinant plasmids. Southern blot. This technique is used to transfer DNA in an agarose gel to a nitrocellulose filter. The basic steps of the Southern blot procedure are: DNA is digested with restriction enzymes, and the resulting fragments are separated by agarose gel electrophoresis. DNA bands within the gel are transferred to a nitrocellulose filter. Next, the DNA is allowed to bind to the nitrocellulose filters by baking at 80 degrees centigrade. The nitrocellulose filter is hybridized with radioactive probes to detect the hybridizing DNA species. Shown here is a Southern blot of an agarose gel containing electrophorically separated DNA fragments. Inside a large empty tray, a glass plate and support are placed. An appropriate amount of electrophoresis buffer is then added. An appropriate filter paper, such as Whatman 3mm filter paper, is placed on top of the glass plate, which will act as a wick for the buffer. On top of the filter paper, an electrophoresis gel containing the DNA bands is placed. Next, a wet piece of nitrocellulose paper is laid upon the agarose gel, followed by a stack of filter papers and soaking pads. Finally, an appropriate amount of weight is placed on top of the pads. This combination is left overnight for the transfer of DNA to the nitrocellulose paper. It normally takes about 8 to 12 hours for the transfer of DNA to the nitrocellulose. The objective of this procedure is to allow the buffer to flow from the reservoir through the gel so that the DNA fragments are extracted and are deposited onto the nitrocellulose. Here, you can see that the bands of DNA have been transferred to the nitrocellulose. Later, this nitrocellulose is used for hybridization experiments. Here we present the live footage of the Southern blot procedure. Here, you can see an empty tray in which an appropriate amount of buffer is added. This buffer normally contains high concentrations of sodium. An agarose gel containing the DNA fragments is placed on the top of a wet filter paper. Normally, Whatman 3mm filter paper is used for this purpose, which acts as a wick during the capillary action. Next, a nitrocellulose paper is placed on top of this gel. The size of the nitrocellulose paper should be the same as that of the gel. Otherwise, an uneven flow of the liquid will cause improper transfer of the DNA bands to nitrocellulose paper. Soaking pads are placed on top of the nitrocellulose. These pads assist in drawing the liquid through the gel. Finally, proper weight is placed on top to compress the pads. Normally, it takes 8 to 10 hours for the complete transfer of DNA bands to the nitrocellulose paper. Finally, the nitrocellulose paper is removed and is used for the DNA hybridization experiments. Special notes. The Southern blot technique is extremely useful to analyze DNA from mammalian genes, plasmids, cosmids, and bacteriophages. As little as 1 picogram of DNA can be detected by this method. Now we present the two most common applications of Southern blot. Southern blot can be used to determine the difference between different double-stranded DNA molecules. On the left, you see a normal piece of DNA. This DNA is cut into pieces using restriction enzymes. These fragments are loaded onto an agarose gel for electrophoresis. In this example, you see 4 fragments as a result of this enzymatic digestion. Now suppose you have another piece of DNA which contains a piece of foreign DNA. You can determine the position of this insert by using the Southern blot procedure. Restriction enzyme digestion is performed in the same way. These fragments are again loaded onto an agarose gel for electrophoresis. Once this procedure is complete, you will be able to locate the foreign DNA insert by comparing the 2 gels and viewing the band shift. After Southern blot, hybridization, and autoradiography, fragments will look similar to the ones shown here. Now we present the second major application of Southern blot. Suppose you have a piece of DNA molecule and a small piece of DNA which can be used to make a radioactive probe. Using the probe DNA, you want to find any similar sequences of probe DNA which might occur in the larger DNA molecule. These particular sequences may occur anywhere within this large piece of DNA. Southern blot is a major tool used to solve this problem. Restriction enzyme digestion of the larger piece of DNA is performed as usual, and Southern blot is set up for these fragments. After Southern blot, hybridization, and autoradiography, suppose you see the middle fragment as a positive match with the radioactive DNA probe. You can conclude that this sequence came from the far left of the larger stretch of DNA. As shown here, the left side portion of the large DNA molecule contains similar sequences of DNA corresponding to the probe DNA. Dot blot is a fast and less time-consuming method for the analysis of specific DNA or RNA species. Basic steps. DNA or RNA samples are directly transferred to nitrocellulose filter. The nitrocellulose filter is baked at 80 degrees centigrade. The spotted filter is hybridized with radioactive labeled probe. Here we show the different parts of the dot blot apparatus. First, a vacuum container to which a vacuum line is attached. On the top of this container, a perforated support plate is placed. Next, a piece of nitrocellulose paper is placed on top of this support plate. Finally, a sample plate is placed where DNA and RNA samples are loaded. A gentle vacuum results in the transfer of these samples directly to the nitrocellulose paper. Now we present the live footage of the dot blot. Here you can see the different parts of the dot blot apparatus. On top of the vacuum chamber, a perforated support plate is placed. Wet nitrocellulose paper, cut to proper size, is placed on top of the Whatman filter paper. Now, a sample plate is placed on top of the nitrocellulose paper. The rubber O-rings at the bottom of the sample plate are used to make the wells airtight to avoid leakage of DNA solutions. The apparatus is clamped and the DNA solutions are loaded into these wells. A gentle vacuum is applied for the slow suction of these DNA samples. Finally, a nitrocellulose paper containing DNA spots is baked at 80 degrees centigrade for DNA hybridization. DNA sequencing. This technique is used to map, verify, and identify the DNA-based sequences of wild-type or recombinant DNA molecules. Enzymes commonly used for DNA sequencing: Clinal, DNA polymerase large fragment; bacteriophage D7 DNA polymerase; Taq DNA polymerase, thermostable; reverse transcriptase, sequinase. DNA sequencing can be performed on either double-stranded or single-stranded plasmid DNA. Here is an example where M13 double-stranded plasmid DNA is used as a vector for DNA subcloning of a foreign piece of DNA. Once this subcloning is complete, the recombinant plasmid is inserted directly into bacteria for the replication of the desired recombinant DNA plasmid. This transfer of plasmid DNA to bacteria is known as plasmid transformation. During bacterial growth, single-stranded M13 DNA is produced, which is isolated and purified from bacterial lysate. This single-stranded DNA, which now contains a foreign DNA insert, is ready for sequencing. A primer DNA, which is basically a synthetic oligonucleotide, is annealed to the complementary sequences of vector DNA. This primer DNA is used for the extension of DNA using various nucleotides, one of which contains a radioactive isotope, which in turn produces an overall radioactive extended DNA. Here, you can see a close-up view of this extended DNA. The green strand is the original single-stranded DNA, and the red strand is the extended radioactive DNA. The DNA sequence of this extended DNA is shown here, which is complementary to the green strand. Note, during your DNA extension reactions, you have also used dideoxynucleotides. These dideoxynucleotides are used to stop the DNA extensions carried by the appropriate sequencing enzyme. For example, here you can see the dideoxynucleotide end positions of the extended DNA. Depending upon the G positions in this piece of extended DNA, there are 4 such end positions: the 2nd, 4th, 5th, and 13th. These fragments of DNA are loaded onto a polyacrylamide gel for electrophoresis. As you can see, the larger DNA fragments stay on top and the smaller DNA fragments migrate to the bottom. Similarly, 3 other lanes corresponding to DDA, DDT, and DDC are shown here. Finally, an autoradiograph is made from this gel, which is used to read the sequence of DNA from the bottom. For example, here you can see the first DNA base is C, then G, next is T, then G, and so on. Normally, you continue to read the gels as far as one can read clearly. This procedure finally provides you with the sequence of the unknown DNA. Now we present actual laboratory footage for the process of sequencing DNA. First, 2 large glass plates are assembled by placing plastic spacers on both the edges so that there is a gap between these plates. These plates are sealed using sealing tape in order to avoid any leakage of the polyacrylamide solution. These sealed plates are then mounted on a support where polyacrylamide gel solution is carefully added. Proper care should be taken to avoid the formation of any air bubbles while pouring the polyacrylamide gel. Before the gel solidifies, a flat spacer is placed on the top portion of the gel. This spacer makes a gap in the gel, which is used later to make the sample wells. It takes approximately 15 minutes for the gel to solidify. The gel solidifies due to the cross-linking agents present in the polyacrylamide gel solution. Once the gel is solidified, it can be used on the same day or the following day for loading DNA samples. Now, the spacer is removed and a comb containing teeth is inserted. This comb makes the appropriate wells for loading DNA samples. These plates are mounted and clamped in DNA sequencing apparatus. An appropriate amount of buffer is added in both the lower and upper chambers. The unit is connected to a power supply and the gel is pre-run until it becomes warm. Before loading the DNA samples on the gel, these samples should be heat denatured. This is done by heating the microfuge tubes containing the DNA samples in both boiling water and then placing these tubes in ice. Approximately 2 to 3 microliters of these samples can be loaded in each well. These samples also contain the blue-colored dye, bromophenol blue, so that the migration of these samples can be checked visually at any time during electrophoresis. This electrophoresis is usually run between 2 and 6 hours, depending upon the separation of DNA bases required. After the completion of the electrophoresis, the plates are removed from the sequencing apparatus. The tooth comb and the top plate are removed carefully so that the gel sticks to the bottom plate. Immediately, a filter paper is placed on the top of this gel, and the paper is lifted carefully so that the gel sticks to the paper. A plastic wrap is placed on top of this wet gel, and it is vacuum dried at 60 degrees centigrade. Once the gel is completely dried, the plastic wrap is removed. This dried gel is exposed overnight for autoradiography by using X-ray film. The exposed gel should contain hundreds of DNA bands, which correspond to different sizes of DNA sequencing fragments. Here you see an example of computer software which can be used for understanding the data obtained by the sequenced DNA. Once you have the sequence of the desired DNA in the computer, you can use this sequence to find out the possible sequence of a protein. This protein sequence is used for understanding the structure and biological function of these molecules. You can also find out the desired restriction enzyme sites present within this piece of DNA. A computerized restriction map of hundreds of restriction enzymes is useful in selecting specific enzymes which can be used to make further subclones or mutations. Special notes. Approximately 600 bases can be sequenced in a single set of sequencing reactions. Computer programs are extremely useful in organizing and analyzing the sequencing data. Mutation. A change in a gene or chromosome is called mutation. Occasionally, replication mistakes or environmental factors cause such mutations. Point mutation. One base of the DNA is substituted, lost, or added. Even changing one base can cause major consequences. Lethal mutations. Mutations that cause death in offspring are called lethal mutations. Mutant. An organism in which a mutation is expressed is called a mutant. Mutagen. Anything that causes or increases the rate of mutations is called a mutagen. Examples: UV rays, X-rays, and other radiations, tobacco, smoke, smog, etc., certain viruses, chemicals, and drugs. Man-made mutations can be introduced at desired locations in DNA. Such mutants are used to study structural integrity and biological functions of proteins, gene expression. Oligonucleotide-directed mutagenesis is normally performed using single-strand DNA vectors. In the example presented here, we want to change the DNA base A to the base C. To change this base, you select a primer DNA containing a mismatch. In this case, G instead of the normal base, T. Here, you can see the incorporation of the mismatch base G into the newly synthesized DNA. Now, the internal DNA molecule is cut with an appropriate restriction enzyme and then filled again. Finally, you can see that the desired mutation is made, in which DNA base A has been changed to DNA base C. There are other mutations that can also be carried out by using similar procedures, such as deletion, in which a piece of the DNA can be deleted, and insertion, where a piece of the DNA can be inserted. Now we would like to illustrate a synonymous example that how changing even one base in a DNA sequence can have major consequences to the DNA. Notice that in this example, we have a group of triplets in the right sequence, as the big fat cat ate the rat. Suppose you remove just one of the letters from this sequence and then rearrange it in the form of triplets. Now you can see that the sequence is frame shifted. Similarly, such mutations can occur in normal DNA molecules. PCR, polymerase chain reactions. PCR is a relatively new technique which is useful in amplifying genes. Basic steps. Two different oligonucleotides are used as primers. The cycle of DNA annealing and DNA synthesis is repeated several times using thermostable DNA polymerase. Applications: to detect rare DNA sequences from pathological and clinical organisms, genetic identification such as DNA from blood, hair, or sperm, etc. This technique is also used by police for crime analysis, to prepare large DNA for cloning, generation of cDNA libraries, to make and analyze DNA mutations. The first step in polymerase chain reaction is to take a double-stranded DNA and separate its strands by heat denaturation, thereby creating two single strands of DNA. Once the DNA is denatured to a single-strand DNA molecule, a synthetic oligonucleotide is used as a primer to extend one of the single-strand DNA from its 5' to its 3' end. Such DNA extension is carried out using thermostable polymerase enzyme, which can work even at 90 degrees centigrade. This double-strand DNA, which was produced by the primer DNA, is again denatured to get single strands. Now you take a second primer DNA, which anneals to the other end of the single-strand DNA. DNA extension is again performed using the same thermostable enzyme, which results in the formation of another molecule of DNA. This procedure of denaturation, primer annealing, and DNA extension is repeated over and over to produce thousands of replicas of the original DNA molecule. A million-fold amplification of the desired DNA is possible in about 50 of these cycles. Footprinting. This technique is used to examine the site of protein-DNA interactions. The portion of DNA bound to proteins becomes resistant to partial digestion by DNase 1. Proteins are allowed to bind to DNA molecules under appropriate conditions. Fragments of DNA are separated on sequencing gel for mobility shift analysis. The first step in DNase footprinting is to label one end of the DNA with a radioactive isotope. After labeling, restriction enzyme digestion is performed using an appropriate restriction enzyme. On the left, you see a normal piece of DNA. Enzymatic digestion of this DNA produces 4 fragments. These fragments are loaded on an agarose gel for electrophoresis. On the right, you see a similar piece of DNA, but some of its restriction sites are now blocked due to the protein binding with the DNA. Enzymatic digestion is performed using the same restriction enzyme. As expected, you are left with only 2 fragments. The missing fragments of this gel are a clear indication of the stretch of DNA that was blocked by the proteins. Nuclease S1 protection assay. Nuclease S1 assay is used to map the 5' and 3' termini of RNA and to locate the position of introns of RNA molecules. Basic steps. DNA-RNA hybrids are made. DNA hybridized to RNA becomes resistant to partial digestion by nuclease S1. Non-hybridized DNA is hydrolysed with nuclease S1. The size of protected DNA fragments are detected under native and denaturing conditions. Electrophorically separated fragments are identified by southern blot. These fragments are used to map the termini of RNA and to locate the position of introns. During the process of RNA synthesis, double-stranded DNA is separated to produce single-stranded DNA molecules. A single strand of genomic DNA takes part in the formation of pre-RNA. This process is known as transcription. Once the pre-RNA is formed, RNA processing occurs in a cell system. the cell system, portions of the RNA called introns are deleted. This process is also called splicing. The final RNA is now referred to as a spliced RNA or messenger RNA. Here we show an example of hybridization between genomic DNA and messenger RNA. Genomic DNA carries both the introns and exons portions, while messenger RNA carries only the exons. During hybridization, DNA carrying the exons portions aligns with the RNA, and the rest of the DNA, which contains introns, is cleaved by the enzyme nuclease S1. Such hybridized samples are loaded on neutral and alkaline gels for electrophoresis and southern blot. In a neutral gel, the DNA and the RNA will migrate as a single band. If you run the DNA in an alkaline gel, the RNA is degraded and DNA fragments get separated depending upon their size. This procedure can also be done at higher temperatures, which cause nicks in the RNA corresponding to the nicks in the DNA. Again, gel electrophoresis is carried out for the separation of various DNA and RNA fragments. The location and size of these fragments can be used to map the RNA molecule. Polyacrylamide gel electrophoresis. This technique is used to resolve the individual proteins from a heterogeneous mixture of proteins. Mixture of proteins are loaded on a polyacrylamide gel for electrophoresis. Proteins are separated based upon their sizes. Smaller fragments move faster than larger ones. The first step in polyacrylamide gel electrophoresis is to assemble two glass plates using spacers so that there's a gap between the two plates. An appropriate size comb is selected, which is used to make sample wells. These plates are filled with a gel solution, which contains acrylamide and bisacrylamide. Before the gel solidifies, the comb is inserted. The process of solidification can take 15 minutes to 1 hour, depending upon the concentration of cross-linking agents. Once the gel is solidified, the comb is removed. Next, the protein samples are loaded into the wells. Here we have shown blue-colored loading samples. In fact, samples look blue when loaded, as they are mixed with a blue-colored dye called bromophenol blue. During electrophoresis, larger molecules remain on top, and the smaller ones move to the bottom. Now we present the live footage of the various steps involved in polyacrylamide gel electrophoresis. Here you can see the assembly of the glass plates, which are used for the pouring of the acrylamide solution. Both sides of these plates are sealed properly using plastic spacers, grease, and clamps. These plates are mounted properly on its support to avoid leakage of the gel solution. These mounted plates are leveled properly to make sure that these plates are in exact vertical position. Next, these plates are filled with separating gel solution, which contains acrylamide and cross-linking agents such as ammonium persulfate and timid. Once this separating gel is solidified, a stacking solution is poured. During electrophoresis, protein samples first concentrate in their zone called a stack, and then they enter into the separating gel, where they separate based upon their size or charge. A comb is inserted to make the sample wells. Protein samples are mixed with an appropriate dye and then loaded on these wells for electrophoresis. Plates containing loaded samples are then placed into the buffer chamber. An appropriate amount of buffer is added, which is stirred slowly during the electrophoresis by using a magnetic stirrer. Special notes. Unlike agarose gel electrophoresis, polyacrylamide gel electrophoresis is commonly run in the vertical position. These gels can range from 10 centimeters to 100 centimeters, depending on the separation required. Caution: acrylamide and bisacrylamide are potent neurotoxins. Proper care should be taken to handle these reagents. Western blot. This technique is used to identify and quantitate specific proteins in a complex mixture of proteins. It is similar to the southern blot, except that proteins are transferred to nitrocellulose. Electrophoretically separated proteins are transferred from gel to a solid support, such as nitrocellulose filter. Transferred proteins are analyzed, usually by antibodies, which react with antigenic epitopes of specific proteins. Proteins immobilized on nitrocellulose filter can also be directly stained with several dyes. Now we illustrate the basic steps in Western blot. You start with a polyacrylamide gel, which contains electrophorically separated protein molecules. On one side of the gel, a nitrocellulose paper is placed. This is where the proteins get deposited during electrophoresis. On both sides of the gel, Whatman 3 mm filter paper is placed. Finally, pads are placed on both sides as well. Normally, electrophoresis containers are built with electrodes for the flow of current during electrophoresis. Before the current is applied, the container is filled with electrophoresis buffer. Current is applied, and the electrophoresis is run at a constant voltage. This process, the transfer of proteins from the polyacrylamide gel to the nitrocellulose, normally takes 4 to 6 hours. Once the proteins have been transferred, the nitrocellulose is removed, and later it is used for antibody binding. First, a primary antibody is used. This antibody will bind to specific proteins bound to the nitrocellulose paper. Next, you select a secondary antibody, which is an enzyme-linked antibody. This antibody has an affinity for the primary antibody. Therefore, it binds to it. Now, an appropriate substrate is used, which reacts to the enzyme-linked secondary antibody. This enzyme changes the color of the substrate, which can be easily visualized. This substrate finally precipitates to the nitrocellulose paper. There are different types of substrates which change their colors depending upon the type of the enzymes. This process eventually allows you to identify specific proteins bound to nitrocellulose paper. Now we present the live footage of the Western blot. Once the polyacrylamide gel electrophoresis is complete, glass plates containing the gel are removed as shown here. The polyacrylamide gel containing the separated proteins is then placed on a nitrocellulose paper. A filter paper is placed on top of this gel. Finally, a pad is placed on top of this filter paper. The other side of the gel already has a filter paper and a pad. This combination is placed in a transfer chamber containing an appropriate buffer. Finally, a power supply is attached, and at the appropriate voltage, proteins are allowed to transfer from polyacrylamide gel to the nitrocellulose paper.